Dr. Aschner is a professor of pediatrics, pharmacology, and senior investigator at the Kennedy Center for Research on Human Development. Dr. Aschner's research interest is on the interaction between genetic and the environment in triggering diseases both during CNS development and senescence. He has been a member or on the board of many organizations and review committees with numerous peer-reviewed publications. Please give a warm welcome to Michael Aschner.
Thank you. Hang on. I want this off just for the Chinese.
Keep it.
You mean on the PhD? Is that what's going on?
I'll tell you later what it stands for. Thank you. Good morning, everyone. I guess I have to read this first. I do not have any financial interest of a product in my talk or with any companies offering grant monies for this continuing dental and medical education program. Okay, so this is going to be, I think, a little bit different. I'm going to talk to you about worms today. I couldn't hear the introduction, but I'm not actually, I don't know if Vanderbilt was mentioned or not, but as of Tuesday, actually, I moved to New York. So I'm sorry I can be here only for a couple of days. I just arrived last evening, and I have to leave tomorrow morning. My lab just arrived on Tuesday to New York, and we're unpacking. So this is my new affiliation, and I've worked with mercury actually from 1979. In different species, and as of about 7 or 8 years ago, I became a worm doctor. So I'll talk to you about some of the studies that we've been doing. Most of it is actually with methylmercury, but hopefully you'll find it relevant to the work that you're doing. So the learning objectives for my lecture are to understand the interaction between genes, loss of function, or knockout of different genes, and the toxicity of mercury and its distribution. I think for those of us that are doing a lot of research, there's a great push to move away from mammalian systems and to find alternative animal models and reduce the number of rodents that we use. So I'll talk to you about some complementary models. And I'll spend most of my time describing to you stress-related transcriptional activation of different enzymes that regulate and that might be protective from the toxicity of mercury. So I don't need to say very much here, but just as a refresher, all of you are interested primarily in mercury vapor. Dr. Adams mentioned already thimerosals in the vaccines. He also mentioned the methylmercury, which is primarily derived from seafood, and I will focus primarily on methylmercury. It's important, though, to remember that some of the methylmercury, which is the organic form of mercury, also tends to get demethylated in various tissues. That happens also in the brain. So no matter what type of mercury actually one is exposed to, there's over time accumulation of the inorganic mercury. And there is a debate in the literature as to whether it's the inorganic mercury or the organic mercury actually that is the ultimate toxic species. So my talk is divided into 4 or 5 or 6, 5 I guess, different bullets. First of all, I'll introduce you to the system that I use, which is a nematode, a worm. I'll talk to you about the transport of mercury. There's less concern, obviously, about the transport of mercury vapor in terms of how it gets into different tissues, because mercury vapor is diffusible, so it gets very readily across different membranes. But the organic form and the inorganic form, either mercuric mercury or methylated mercury, needs to get into different tissues, and to do so it requires various transporters. So I'll talk to you about the amino acid transporters which shuttle the methylmercury. I'll talk to you about early life exposure to methylmercury in wild type animals and in animals that have a deficiency in an enzyme or a protein that has been associated with Parkinson's disease, and I think it will become clearer as we get to that point. I'll talk to you also about the role of what's known as the master switch for regulating antioxidant responses in mammalian systems and in invertebrates, which is known as SKN1, and how that might be related to toxicity and specifically affect areas that are known to be affected in Parkinson's disease, namely in dopaminergic neurons. And then if we have time, I'll talk to you about some additional developmental effects of mercury. So this is the system that we use in my lab. These are worms. They're about 1 millimeter in size. They're very tiny, but you can see them in a naked eye. We use them because they're very easy to culture. What they eat is bacteria. They eat E. coli. And the strength of this system is because we can use very readily genetics. We can use what's known as interference RNA, and we can silence specific genes. So we can ask questions about what's the function of a given gene within this animal model. And also, many of the genes that are expressed in humans are not expressed in the nematode. They have only about 19,000 genes. We have about 34,000 genes. So we can take specific genes, we can put them in the worms, and then we can ask specific questions about what's the function of that gene product within the nematode, and presumably extrapolate to mammalian systems. They have only 959 cells. And for those of us that are interested in neuroscience, the advantage and disadvantage at the same time is that they have only 302 neurons. And actually, a Nobel Prize was given to Sidney Brenner for doing a lot of the work in the nematode. And basically, the lineage of every individual cell is known in terms of when it's being differentiated, where it migrates, when it stops migrating, when it dies, and so forth. So we know a lot about this system. As I said, they have about 19,000 genes. We have about 34,000. Another strength of the system is that, obviously, if you're interested in mammalian genes, there has to be a great homology between the gene that you're interested in humans and the gene in the nematode. If there's very low, homology between mammalian systems and the nematode, then the question that you're asking, you might not be getting the right answer because of it. So in general, there's about 70% homology between mammalian genes and invertebrate genes. And as I said before, some of the genes that we have are not expressed in C. elegans, so we can put these genes into the C. elegans and ask what's their function and extrapolate to mammalian systems. I'm going to skip some of these because I won't have enough time. So the first question is, how does methylmercury get into different tissues, and how can we look at it by using this model system? So we've done some studies, and others have done some studies showing that in humans, the large amino acid transporter, LAT1, is important in shuttling methylmercury into the brain. In C. elegans, there are 9 different genes that are encoding amino acid transporters, which are similar to the LAT1, and they're known as AAT1 through 9. I'll focus on my talk, during my talk, only on AAT1 through AAT3, because these have the highest homology between mammalian systems and invertebrates. And also, these are the ones that have the cysteine, which is responsible for the disulfide bridge that exists between the heavy and the light chain of this transporter. I'm not going to talk about this, so I'm going to skip it. So basically, the question that we're asking here is, what is the function of the AAT transporters, and is it a mechanism that is shared between mammalian systems and the invertebrates? And what happens if we knock out these specific transporters? Are the animals going to be more protected against methylmercury toxicity? That is, if they don't have a system to transport the methylmercury, they should be more resistant to its effect. I know you can't see it, but the purpose of this is just to show you that there's great homology between these transporters. In each of these lines or panels, there's AAT1, AAT2, and AAT3. The conserved identical amino acids residues are shown in green, and the homologous residues are shown in yellow. So you can see that there's a great homology between AAT1, 2, and 3, and LAT1, which is the mammalian system for the transport of methylmercury. So the first question that we asked was, what happens if we take the worms and we treat them with a substrate that we know is transported by the neutral amino acid transporter, by LAT1 or AAT? I should back up and say that the way that methylmercury gets into tissues is by conjugating with cysteine. And if you look at the structure of the cysteine and methylmercury conjugate, it's very similar to the structure of methionine. So it's actually been coined molecular mimicry, is a term that refers to the fact that the methylmercury cysteine conjugate can mimic the structure of methionine, and if you wish, it fools the transport system, leading it to believe that rather than transporting methionine, it transports methylmercury cysteine. So if you take excess of methionine, what you should be able to do in this case is to decrease the uptake of methylmercury into tissues. And this is exactly what we've done. We've treated the animals with a methionine, pre-treated them with methionine, we've given them methylmercury, and then we looked at the LD50, which is the lethal dose or lethal concentration that kills 50% of the animals. So this is a survival rate on the y-axis. This is the log dose of the methylmercury. These are the wild-type animals shown over here. And you can see as you increase the concentration of mercury, more of the animals die. If the concentration is high, all of them essentially die. The LD50 is right here, so it's the 50% dose that kills half of the animals, the dose that kills half of the animals. And you can see that if you give them methionine, you have a rightwardly shift in this survival curve, which means that the methionine protects the animals from the methylmercury. So that was the first hint that, LAT1 or the AATs are probably responsible for mercury transport also in the nematode. The next thing we did, and this comes back to what I've said before, it's very easy to knock out genes in the nematode. What we have to do is take a double-stranded RNA, we give it to the bacteria, the worms eat the bacteria, you come back a few hours later, and the gene product is not expressed anymore. So we could do those studies actually within days rather than years if we were to use mice or other animals. So just for orientation, here we look at the RNA levels for the different transporters after treating the animals with the double-stranded RNA against AAT1, which is shown here in A, AAT2, which is shown in B, in A, and AAT3, which is shown in C. Now, if you remember from the green and yellow slide, there's great homology between AAT1, 2, and 3. So it's not really surprising that when we use the RNA, interference RNA, against those different transporters, that we actually knock down the RNA levels for all three of them. So if we use the double-stranded RNA just for AAT1, we knock AAT1 down, AAT2 and AAT3. We do pretty much the same thing for AAT2, and we get the same thing for AAT3 with the exception of AAT2. The reason for that is that there's, again, great homology between the three different isoforms of the AATs, of the transporter. Now, if we knock out these transporters and then we look at survival, this is AAT1, this is AAT2, and this is AAT3. I'm not going to talk about D. What you could see is that when we knock out the transporter AAT1, there's a rightwardly shift in the sensitivity of the animal. So for the same dose, less animals are dying. You see the same thing for AAT2, and you don't see any difference for AAT3. So it seems that AAT1 and AAT2 are the predominant transporters for methylmercury in the worm. We also looked at the levels of mercury in these animals. So these are wild types animals. These are the levels, these are the concentrations of mercury. And you can see a dose-dependent increase in the worms. When you knock the transporter, AAT1, AAT2, and AAT3, for all three of them, you actually see a decrease in the amount of mercury compared to what you see in the wild type. And this is true for each of the given doses within each of these panels. So from this part, we can conclude that AAT1 through AAT3 are homologous to each other, and they result in multiple gene knockdowns in RNAi-fed worms. AAT3 knockout attenuated methylmercury accumulation in the worm. AAT1 is the most homologous to the neutral amino acid transporter, the large neutral amino acid transporter, LAT1, and was knocked down in all three RNAi feedings, explaining the lack of differential uptake across the three RNAi-fed worms. So collectively, these studies support a conserved role for LAT1 in methylmercury transport in the worm, and methylmercury toxicity was dependent specifically on AAT1 through AAT3. Okay, I'm going to talk next about the impact of early life exposure to methylmercury on different toxic outcomes within the context of animals that are wild type and animals that have a mutation or knockout of a gene that's associated with Parkinson's disease. So this is a map of the prevalence of Parkinson's disease in the United States. And if Parkinson's disease was only a genetic disease, you would expect to see an homologous distribution of the disease. That is, it should be similar in New York to Los Angeles and Miami. It's not. About 80 to 90% of PD is not attributable to genetics, with the exception of the familial type of Parkinson's disease, which usually tends to develop in individuals at a very young age. Michael J. Fox would be one example. The sporadic or idiopathic Parkinson's disease, don't know what happened. Sporadic or idiopathic Parkinson's disease is only about 10% genetic. And you could see actually, and I was surprised when I saw that, because given the fact that pesticides are known to induce or correlate with Parkinson's disease and many other compounds, I would have expected to see the highest incidence or prevalence of PD in the Midwest, but that's not the case. There are many options for this, or many possibilities for this type of map, and we can talk about it later. But the point of it is just to make you aware of the fact that there is an environmental or many environmental components to the sporadic or idiopathic Parkinson's disease. So 10 to 20% of PD, as I said, has genetic origin, other than the familial type of Parkinson's disease. As far as mercury and Parkinson's disease, a causal relationship between mercury and PD has been noted. Both Parkinson's disease and mercury poisoning have some similar symptomology, both sensory, which is the initial part of PD and is also expressed in mercury toxicity, and also some of the motor functions that are seen, or dysfunction that are seen in both diseases. Several studies also support an association between fish consumption or occupational exposure, including dentistry, to mercury compounds, an increased prevalence of Parkinson's disease. And as noted in the last bullet, low serum mercury levels are associated with decreased risk for Parkinson's disease, and serum mercury levels are significantly higher in post versus pre-Parkinson diagnosis. Now, the gene that we're interested in is known as Parkin in mammalian system. You may have heard about it, Park2 or Parkin. I don't know why it's always so confusing, but the nematode has a different name, so it's PDR1. But PDR1 is Parkin, and I'll exchange those names as we go along. PDR1 is the homolog of PARK2 and PARKIN. I'll show you its function in one of the next slides. So the question that we had here is, what is the interaction basically between exposure to mercury and loss of function mutation in this gene? And the hypothesis was that early life exposure to methylmercury and knockout of PDR1 or PARKIN will exacerbate the toxicity of mercury and damage dopaminergic neurons. Dopamine is the neurotransmitter that's mostly affected in Parkinson's disease, and I'll show you some of the studies that we've done to address this issue. So this is the function of PARK1, of PARK2 or PDR1. What it does basically, it's an E3 ligase. It takes different proteins and it ubiquitinates them, and as they get ubiquitinated, those target proteins then are being shuttled to the proteasome and they're degraded. And this is one way where the cell can recycle basically different types of proteins. So the hypothesis was essentially that if you have an abnormal gene, if you have an abnormal E3 ligase, that some of the transporters from mercury may not be shuttled to the proteasome. You may get increased expression of this protein, and this may lead to exacerbated toxicity in worms or individuals that have a mutation or loss of function in these genes. So the first slide is again looking at the lethality in these animals. What we have is the number of live worms, looking at the dose of mercury. N2 are the wild types, so these are normal worms. As you increase the concentration of mercury, more animals are dying. And you can see in the PDR or PARKIN knockouts, there's an enhanced sensitivity. So animals that have a knockdown of these genes for the same amount of mercury show increased lethality, or you get less live worms. If you look at their survival over time, these are the control, the wild type worms. As you increase the concentration of mercury, as you expect, more worms are dying. So there's again a leftwardly shift. The first thing that you could see that the animals that have a knockdown of the PDR actually live about 5 days shorter compared to the wild types. And then for the same dose of mercury, you get a much greater effect in terms of their survival. And you also get a gene by a strain interaction. If you look at the number, the brood size for these animals, what you find out is that mercury itself in the wild type decreases the brood size, and this effect is exacerbated in the PDR knockouts. Again, you see a dose effect, a strain effect, and in this case there's no dose by strain interaction. The point again is that the PDR knockouts are much more sensitive to the mercury compared to the wild type animals, and you see it with a high dose and with a low dose of mercury that we use. If you look at the amount of mercury that accumulates in these animals using ICP-MS, as you expect, the more mercury the worms get, the more mercury you find in the worms. This is true in the wild types, which are shown in black, and you could see that the PDR knockouts have significantly more at each of the doses compared to the wild type animals. And as we hypothesized, what we find is that in the PDR knockouts, there seems to be enhanced expression of the transporter. So here we're looking at AAT1, AAT2, and AAT3 expression. This is the transporter expression by looking at RNA levels in the worms. You could see that with increased mercury, we get increased expression in the wild types, but this effect is significantly increased in the PDR. Again, suggesting that in the animals that don't have this gene expressed or the gene byproduct expressed, there's a dysfunction and disability to recycle, if you wish, the transporter. It does not get to the proteasome and it does not get degraded. So one of the mechanisms by which mercury affects cells in brain and other tissues is by inducing reactive oxygen species, and I'll talk about it in more detail a little bit later. What's interesting is that we actually found that in both PDR knockouts and the wild types, the mercury causes an increase, especially at the low dose, in the amount of reactive oxygen species, but there was no difference between the PDR knockout and the N2. So it seems that other than increased oxidative stress, there must be other mechanisms which account for the change that we're seeing in the survival and lethality of methylmercury in the PDR knockouts compared to the wild types. This is the system that regulates oxidative stress. It's known as NRF2 in mammalian system. In the nematode, it's known as the SKN1. And essentially, to enhance the expression of antioxidant enzymes and macromolecule, what happens is that when you have oxidative stress within the cell, there's a protein which is known as KEAP1, which has a bunch of cysteines on it. The increased oxidative stress causes the oxidation of these cysteines. That causes the KEAP1 to separate from the NRF2, and then the NRF2 is free to move into the nucleus, where it binds to a DNA consensus site, which is known as the antioxidant response element. What you get once the NRF2 binds to the ARE is upregulation of phase 2 detoxifying enzymes, and the cells start making a lot of antioxidant stress proteins. GST4, which is part of the glutathione system, hemoxygenase, and many other proteins. So here we looked at the ability of these worms. Again, we're still talking about PDR1 knockouts to respond to methylmercury. And we did this again in the N2, the wild type animals, and the PDR knockouts. And what you can see is that compared to the wild type animals, which show a very nice response after they've been treated with methylmercury, this response is significantly attenuated in the animals that have this gene knocked down, the PDR knockout. So it seems that these animals are not able to mount an appropriate antioxidant defense response subsequent to their exposure to methylmercury. And finally, for this part, we can also look at specific behaviors in the nematode. I'm not going to show you a lot of detail, but we know that the dopaminergic neurons, which are the ones, again, that are affected in Parkinson's disease, are important in seeking behaviors, in food-seeking behavior in the nematode. So when you knock down dopamine, the animals can actually survive, but they cannot reproduce, and they do not eat as much as the wild-type animals. So what we do here is we look at a behavioral paradigm in the worms, in the wild-type worms, and in the PDR knockout animals. What we do is we look at body bends, which is an indication of food-seeking behavior, and we calculate the ratio of animals that are off food and on food. So each animal actually is being used for its own control. And you could see that the number, the change in the body bends, and this is in 20 seconds, is about 5 for the wild types. If you treat the animals with mercury, they seek less food, their movement goes down. These are animals that have been treated with 10 micromolars of methylmercury and 20 micromolars of methylmercury. You could see that the animals where we've knocked out the PDR1 actually look like animals that have been intoxicated with methylmercury, but when we give them additional mercury, they actually do not go further down compared to their respective controls. And finally, we can confirm this by looking at animals that don't make dopamine at all. We've generated some strains that don't have tyrosine hydroxylase, so they don't have the ability to make dopamine. And you could see that these animals look pretty much the same compared to the animals that are PDR1 knockout. So this suggests that methylmercury in wild-type animals, and especially in the PDR knockout strain, significantly affect the dopaminergic system. And I'll show you some more data about this a little bit later. So in conclusion of this part, we can say that the combination of early life exposure to mercury and Parkin-2 knockout or PDR knockout have significant effects on oxidative stress and aging. In response to mercury exposure, PDR1 knockout shows similar levels of reactive oxygen species, but nevertheless, they do show an altered or dysfunctional ability to respond to oxidative stress because their SKN upregulation, as I've shown you in one of the slides, is decreased. Early life methylmercury exposure is a risk factor for loss of dopaminergic function later in life in wild-type worms. However, the combination of PDR knockout and early life methylmercury does not further exacerbate the already reduced dopaminergic function, which is produced by knockdown of the gene itself. Okay, if you recall, I've shown you the scheme about SKN1, which is the master regulator of antioxidant responses in mammalian system and invertebrates. So I'll show you some studies now on the role of SKN1 in methylmercury-induced latent dopaminergic neurodegeneration. So as I said before, SKN1 is a major stress-activated cytoprotective transcription factor. It promotes transcription of different enzymes that scavenge free radicals, synthesize glutathione, and catalyze reductions or reactions that increase xenobiotic excretion. And the hypothesis of this next part of my talk is that the extent of methylmercury toxicity is going to be dependent on the ability of these animals, to respond to an intact SKN1 response. So if these are wild-type animals, they're going to be less resistant to the effect of methylmercury, whereas SKN1 knockouts are going to be more sensitive because they're going to have a deficient ability to respond to methylmercury. To do this, we generated a number of strains. We knocked down SKN1. Again, this is done by using bacteria with double-stranded RNA, which can interfere with the SKN1. We've also generated another strain where we can hook up a green fluorescent protein to the promoter of SKN1, so we can look at these animals under the microscope and see the expression level of the SKN1. I should have mentioned before that these animals are actually transparent, so you don't have to section them. You could just do those kinds of things and then just look under a fluorescent microscope and see exactly where the fluorescence is. So the N2 again are the wild-type animals. As you increase the dose of mercury, you get increased lethality. And if you knock out SKN1, you can see that these animals are much more sensitive. So if you do not have an ability to respond to oxidative stress, you are much more sensitive to methylmercury, and this is due to the effect that you cannot generate appropriate levels of glutathione, GST4, heme oxygenase, and other proteins that are associated with antioxidant defenses. We've looked at mercury content in these animals, and we found a significant effect of dose on mercury content. This again is measured by ICP-MS, but the mercury content was indistinguishable between the N2 wild type and the SKN1 knockout. There is somewhat of a trend for an increased concentrations of mercury in these animals, but it's not significant. When we look at survival, we find that as you increase the concentration of mercury in the wild type, this is shown in black, you get a leftwardly shift as you'd expect. But you could see that the SKN1 animals are much more significantly affected. There's a further shift to the left, meaning that for a given concentration or dose of methylmercury, the SKN1 knockouts are much more sensitive than the wild types. We've looked also at reactive oxygen species. As you would expect, the number or the amount of oxygen species is increased in N2 animals. There's a slight increase. and you get a much greater amount of oxidative stress in the animals that don't have the capacity to respond to the induced reactive oxygen species. So comparing these two bars and these two bars. And this is, I don't know how well you can see it, but these are the SKN1 green fluorescent protein worms. These are control animals, so you get some fluorescence in these animals. These are the animals that have been treated with methylmercury at 10 micromolar, and you could see, especially at 20 micromolar, there's a significant activation of the SKN1 system, suggesting that these animals are trying to cope with the increased oxidative stress. We also looked at dopamine levels in these animals. These were measured by high-performance liquid chromatography immediately following 30-minute exposure to mercury. And as you can see, there's a significant difference between the 0 micromolar methylmercury wild-type worms and all the other worms. You could see that the amount of dopamine in the animals that are the knockouts is significantly reduced compared to the animals that are the wild types. Again, pointing out to the fact that the dopaminergic system is more sensitive to methylmercury if you have a mutation or loss of function in this gene. And we've looked at some behaviors, and I don't know why it's not showing here. It was showing on my Mac, but basically this shows what I've shown you before, that when you don't have SKN1, just like when you don't have PDR, the number of bends, the difference in bends, body bends between off and on food is significantly attenuated in the animals that don't express SKN1, and it looks similar to what you find in animals that don't make any dopamine at all. Again, pointing to the fact that the dopaminergic system is sensitive to the methylmercury. I'm going to go to the next slide, and I don't know if it'll work, but okay. So what you're looking at here are live worms in a movie. What we do is we can put a green fluorescent protein in the promoter for a protein that's expressed only in dopaminergic neurons. This is a green fluorescent protein. We put in the promoter that's expressed only in dopaminergic neurons. The protein is known as the dopamine transporter. And when we do this, we can label only the neurons that express the protein. And if you look under the microscope, those proteins are going to look, or those cells are going to look green. So you'll have to believe me, there's 6 of them in the head, and there's a couple of them in the tail. The 6 in the brain are for thinking, the 2 in the brain are for the reproduction. And I'll show it again. So this is the worm. It's about 1 millimeter in size. As I said, it's transparent. These are the 6 neurons in the head that are labeled with green fluorescent protein for the dopamine transporter, and there's a couple of them in the tail, which you'll see come and go. So we can ask specific questions about mercury, about manganese, about any compound that you might be interested in that might be found in the dental amalgams or composites, and we can ask specifically, what is this compound doing to the dopaminergic neurons? So these are the same neurons, and now they're stained with cherry red instead of green fluorescent protein. Again, you'll have to believe me, there's 8 of them. 6 of them are in the head. This is the wild type. You could see these neurons when they're not treated with mercury, 0 micromolar. If we treat them with mercury, 20 micromolar, most of these neurons disappear. The fluorescence goes away. If we look at the SKN1 knockout, these are the animals that don't have the ability to respond with an appropriate antioxidant response. You could see that there seems to be, even at basal levels, some alteration in the structure of these neurons. And at 20 micromolar, the effect seems to be significantly greater compared to what we see in the wild types. So in conclusion, mercury content showed no strain-dependent differences. Reactive oxygen species generation was dramatically increased in the SKN knockout compared to the wild-type worms. The structural integrity of the dopaminergic neurons showed loss of neurons in the SKN1 knockout, and it was exacerbated in the presence of methylmercury compared to the wild-type controls. The dopamine levels were decreased in response to methylmercury exposure and in SKN1 knockouts, and functional behavior assays showed similar findings. And together, these studies suggest that knockdown of SKN1 in the worm increases dopaminergic sensitivity to methylmercury exposure. I'm going to show you just a couple more slides, and these are from animals that are actually treated with methylmercury very early on. What I've shown you up to now is in adult animals. So what we've done here is to look at methylmercury treated worms. Basically, they're a lot smaller, and I think you could see it here. These are at the larval stage 1, larval stage 1, 24 and 48 hours, and larval stage 48 hours. If you compare the controls to the methylmercury treated ones, you can see that especially here, they tend to be much smaller. I've showed you before that also the brood size is much smaller in animals that are treated with methylmercury. We can also look at the developmental stages in which these animals are in. There's L1, 2, 3, and 4, and then they turn into adults. And this is the percent of animals, this is the concentration or the dose of mercury, and these are the different stages. And you could see that if you look at these animals with no mercury, that after 48 hours, most of the animals, about 90% of them, or 70%, I'm sorry, are in the L4 stage. If you give them increased concentration of methylmercury, you could see that most of the animals arrest. If you compare this, they're in the L2 stage, whereas in the 0, 70% of them are already in the L4. So it's hard to see, but over here, with a 1 millimole mercury at the 48-hour post-treatment, there's only about 10% versus 70% in the animals that don't get the mercury that are in the L4 stage. So the methylmercury causes developmental delays. We've also looked at the accumulation of mercury at the different stages. This is in L1, larval 1 stage, larval 1 stage, larval 4. This is at 30 minutes. This is L4. This is L4 15-hour treatment. This is 6, 15, and this is in the L4. And you can see within each of these groups, basically, there's more mercury with increased dosing. What's interesting and different actually from vertebrate animals is that we find that there's more mercury accumulating in the adult, in the larval 4 stage, which is a more advanced stage compared to the L1, which is somewhat contradictory to what you find in mammalian systems, where the fetus seems to be most sensitive. We've also looked at some behaviors, and this is just to show the number of pharyngeal pumpings in the worm. That's the way the worm gets its food. And you can see a nice dose-dependent decrease in the number of pumps as you increase the dose of methylmercury from 0 to 0.75 millimolars. The metallthionines are also important in regulating mercury toxicity. These are small proteins. About 30% of the content of the metallthionines is cysteine, which contains sulfhydryls, so it's a good buffer for different mercury species. It's involved in detoxifying different metals. It's also known to protect from oxidative stress because of the SH groups on it. The worms have couple genes, 2 metallthionein genes, 2 isoforms, the metallthionein 1 and the metallthionein 2. This is what happens when you give them methylmercury. This is the metallthionein 1, no mercury. This is metallthionein 1 with 0.4 millimolar of methylmercury. You get slight upregulation, and you see the same thing with metallthionein 2, slight upregulation of the metallthionines. If you knock out the metallethionine, what you see is a leftwardly shift in the sensitivity of the animals. Again, we're looking at survival. The wild-type animals are shown in blue. As you increase the concentration of mercury or the dose, more animals die. If you knock out one of the genes, you get a leftwardly shift in green. This is the MT1 knockout. If you knock MT2 in red, you get also a leftwardly shift. And if you knock both of these genes, the yellow, it looks pretty much like the MT2 knockout. So you don't get a synergistic effect between the two, but clearly the metallethionine knockout makes the animals much more sensitive to mercury. So another consideration other than the PDR knockout, the SKN1 knockout, is the metallethionine knockout. All these genes seem to exacerbate the toxicity of mercury. So the C. elegans, just in summary, is an ideal model for studying interaction between loss of function or gene knockout and methylmercury toxicokinetics and toxicity. As I've shown you in the first slide, we believe that AAT1 to 3 and predominantly AAT1 is important in shuttling methylmercury into the worms. The combination of early life exposure to methylmercury and the knockdown or knockout of PDR1, Parkin2, have significant effects on oxidative stress and aging. The knockdown of SKN1, which is the master switch for regulating antioxidant responses, increases the sensitivity of the worm to methylmercury, and this is especially related to sensitivity of dopaminergic neurons, which, as I mentioned, are important in Parkinson's disease. I hope that I've convinced you that genetic predisposition is a key factor for mediating the effects of methylmercury on neurodegeneration, specifically the dopaminergic system. And in the last part, I showed you that methylmercury toxicity, developmental toxicity, in terms of survival of the larva, the brood size, and so forth, is dependent on the redox status. I didn't show you any data on glutathione, but with metallethionine knockout, the animals seem to be more sensitive. There's increased stress associated with the knockdown of metallethionine, which is expressed by increased lethality. And all of these are related to transcriptional activations of different enzymes and proteins, or the lack thereof, that seem to be affected in these different strains with the different genetic backgrounds. And finally, I want to thank Dr. Fleischer for, I saw him earlier, I think he's in the back. Thanks for inviting me. Hope to spend more time with you in the future. These are the people that did the work. Kirsten Helmke did most of the developmental stuff. Ebony did the work on the PDR knockout. Sam did the work on the AAT, and Alex and Catherine didn't do any mercury work, but they were the first ones to set the C. elegans model in my lab about 7-8 years ago. And the ICP-MS work was done actually in Norway at the University of Trondheim. And last but not least, this work was funded by the NIEHS by an R1 007331. And I'll stop here. I think I'm on time, so I'm happy to answer questions you may have.